variety of living things we see around us often seems endless, and rightly so, since organisms number in the millions. The diversity of life is easily apparent to us all. But with all its diversity, life as a whole is also amazingly unified. The more we study living organisms, the more striking similarities we find. Just as atoms and molecules are the basic units of all existence, they in turn combine to form the smallest basic unit of life as we know it, the cell. Cells in turn multiply and grow to form complicated organisms. In the same way, a cell about the size of a pinhead is the beginning of a human embryo. But however complex their structure, all animals and plants are composed of cells, and all cells have been produced by cell division.
Cells are the smallest living units in your body. They come in all shapes and sizes. This is a live cell scraped from the inside of a cheek. These are cells from cartilage. Nerve cells. Heart muscle cells, each with its own inherent beat. And these are blood cells, rushing through microscopic blood vessels. The larger, slow-moving ones are white blood cells. These are red blood cells. With the invention of the microscope, tiny organisms previously unknown to the study of biology were discovered. Cells were found to be tiny packets of life, dynamic units which showed remarkable diversities, each with certain structures suited to particular functions. But in spite of this diversity, cells were found to have certain characteristics in common. A dark body, soon named the nucleus, was the largest and most easily recognized of the cell's internal structures. Each cell also had an outer membrane, which surrounded all the material of the cell, and inside were tiny organ-like structures. As microscopes improved, further discoveries were made. The cell, it was agreed, was the basic unit of life, and all cells were produced by a seemingly simple and identical process, cell division. In cell division, the nucleus played a central role. It was clear that some substance in the nucleus was vital to the cell. In the early stages of division, this substance was gathered together. Then it was divided exactly in half. This was the hereditary material of the cell, the chromosomes. These and other discoveries unified the study of plants and animals into the single study of biology.
After the discovery of a cell's nucleus, there was nearly a 100-year period of other significant discoveries. More cell parts were discovered. It was found that although cells were different in size and structure, every cell had the same basic parts. A cell is enclosed by a membrane that separates it from the outside world. And within the cell membrane is a jelly-like mixture called the cytoplasm. Another membrane surrounds the nucleus and separates it from the cytoplasm. Different kinds of cells are variations of this same design. The constantly changing shape of an amoeba, the flattened cells of the skin, long, narrow nerve cells, and the trumpet-like shape of the stentor. All cells share these same basic structures. Every cell, whether a tiny organism living in a drop of water or part of a larger animal, has the same basic needs. To survive, it must consume food and get rid of waste materials. Water has been removed from these cells. Left this way, they would soon die. But when water is available, it's quickly taken into the cell. Taking water in and out is the job of the cell membrane. The cell membrane expands as water is taken in. In this way, the cell membrane keeps the cell alive and functioning properly. In plants, the cells are surrounded by an additional structure, the cell wall. The cell wall helps protect plant cells and maintains their shape. Plant cells can also capture energy from the sun and use it to make simple food. This process takes place in tiny green structures in the cytoplasm. Other structures use this food to release energy to the rest of the cell. Cells have many structures, each designed for a specialized function. In spite of the fact that the cell is the smallest living unit, it's a highly complex structure involving many parts and many functions. The functioning parts of a cell are called organelles. The most obvious and visible organelle of the cell is the nucleus, the control center of the cell. The nucleus is bounded by the nuclear membrane. The material inside the nucleus, called chromatin, controls, regulates, and directs all the functions of the cell. And a part of the nucleus which is easily seen is the nucleolus. Its function is largely a mystery, but we do know that it's involved in the production of another organelle for the cytoplasm, the ribosome, which we will discuss later.
Surrounding the entire cell is a membrane which separates the cell from its environment. It regulates all materials that enter and leave the cell. Between the cell membrane and the nuclear membrane is the cytoplasm. Cytoplasm is mostly water and contains all the other organelles. These organelles are directed by the nucleus to do all the things necessary to keep the cell alive and functioning. The cytoplasm and its organelles can be thought of as a factory. All of the cell's actions require energy, and the energy is supplied by an organelle called a mitochondrion. This is the powerhouse of our factory. The production line of our factory is the endoplasmic reticulum, where the materials necessary for the cell's livelihood are produced. The reticulum takes small materials that come through the cell from the outside and assembles them into useful products. Some of these products, like fats and carbohydrates, remain in the cell for its own use. In addition, some cells produce other products like hormones, oils, or digestive enzymes that are exported from the cell to other parts of the body. The ribosomes manufactured in the nucleolus we mentioned earlier are transported through the pores of the nuclear membrane and appear as dots on the surface of the endoplasmic reticulum. There they assemble protein molecules.
Now all of these products manufactured by the ribosomes and endoplasmic reticulum are then transported to the storage and packaging area of our factory, the Golgi body. Here the materials are put into packages called vacuoles. Some of these vacuoles remain in the cell for its own use. Others move to the cell membrane, where they release their contents, such as hormones, to the outside. It's apparent at once that the cell is a living and dynamic structure. These organisms are composed of just one cell. This cell is basically the same as most of the cells of your body. They all have a nucleus, ribosomes, and a cell membrane. The cell membranes are flexible, allowing for movement and the intake of nutrients. But there are some differences in cells. Different shapes, different methods of eating, different numbers of organelles. They secrete different products, and they move in different ways.
What we know about cells, we've learned through the use of a microscope. During this series, we'll be using several kinds of microscopes. In our studio, we have a compound light microscope attached to a studio camera, so you can see at home what I see in here. This light microscope shows you a picture like this. The next step upward in microscopes is the electron microscope, which is used to take photographs called micrographs. This series of slides shows you the difference. Using the light microscope, we'll look more closely at this section between two muscle cells. We can increase the magnification and bring that area into a much better view. But if we increase the magnification again, focusing on just this portion, the picture blurs. Now that same portion through an electron microscope, you can see how much clearer the image is. This, by the way, is a red blood cell inside a blood vessel. We can continue to increase the magnification. We see the dark red cell and the light vessel wall. And finally, just the vessel wall.
The electron microscope is a tool that gave us our current view of the cell as a complex structure. Now then, let's compare the drawings that we saw earlier with the actual cell parts. Here's a nucleus and a nuclear membrane. You can see the pores through which the ribosomes exit. And here's a mitochondrion, the powerhouse of the cell, recognized by the internal membranes. Here's the endoplasmic reticulum, the assembly line for the manufacture of fats and carbohydrates. And the ribosomes, which work on the surface of the endoplasmic reticulum, producing proteins. Here's the Golgi body, the warehouse and packaging area of the cell. The material is pushed to the ends and secretion packages or vacuoles break off. This empty chamber is a vacuole. And here's a structure we didn't discuss before, a lysosome. Lysosomes can be thought of as very specialized vacuoles that contain digestive materials. When an old cell dies, the lysosome bursts, and its material digests the dead cell parts so that a new cell can take its place. So you can see that most of our knowledge of these cell parts has been accumulated in the last half century since the invention of the electron microscope.
An even more recent development in microscopes is the scanning electron microscope. This produces an image with almost a third-dimensional quality. These are red blood cells. You can see the surface and the shape of what you're looking at instead of just a slice. This is the surface of a pearl. Pollen, a cell surface. One-celled animals, bacteria, the beginning of a blood clot, more human cells, and finally, the early stage of an embryo.
We've talked mainly about animal cells. While all cells are very similar, there are a few differences in plant cells. For instance, plant cells don't have the tiny lysosomes we just mentioned, but they do have chloroplasts, which are used in photosynthesis. Probably the main difference between plant and animal cells is the cell wall. It's the cell wall that gives plants the support to stand erect. However, it's the membrane that regulates diffusion. In plant cells, the membrane is inside the cell wall. In an animal, the membrane is the outer part of the cell. because animal cells don't have a cell wall. Now there's one more kind of cell that belongs neither to the plant nor animal kingdom, bacteria. Bacteria fall into a category by themselves. They're much more primitive than the plant and animal cells. They have few internal parts. They have no nuclear membrane, no mitochondria, no vacuoles, and so on. Because they are so different from plant and animal cells, we usually ignore bacteria when discussing the cell theory.
Cell theory simply states that all living things are composed of cells, and that the cell is the smallest unit of life. Another commonly accepted part of the cell theory involves perhaps the most fascinating part of the study of cells. This part states that all cells come from other cells, and that process of cell division is mitosis. In unicellular organisms, like this amoeba, division produces two complete independent amoebas, just like the first one. In multicellular organisms, cell division permits an increase in the number of cells and permits the growth of the entire organism. In other words, each cell is not an independent organism. It's part of the whole. New cells in multicellular organisms are used for growth and repair of tissues. But what exactly is mitosis, or cell division? And how can one cell split into two identical copies of itself?
Although the entire division is one continuous process, biologists have divided mitosis into 4 visible phases during which certain events occur. Between divisions, the cell is in the stage called an interphase. Before any visible change takes place, the cell makes a copy of all of its genetic material. Then, in the first of the 4 visible phases, chromatin condenses into coils, the chromosomes. Soon we can identify them as duplicate sets of chromosomes. The chromosomes then arrange themselves along what's called the cell's equator. The duplicate chromosomes then separate. An identical set moves to each end of the cell. The cell wall gradually forms in the middle, and this creates 2 new daughter cells with the same hereditary characteristics as the original.
Now let's watch mitosis according to its phases. During the interphase period prior to mitosis, the cell has been busy with the functions of its normal life, and in particular, with its own growth. During this period, the chromosomes aren't the rod-like bodies we normally associate with the word. Instead, they're in the form of a twisted mass of thread-like material called chromatin. At the end of interphase, the chromatin content doubles in preparation for division, but we can't see the doubling. Prophase is the first visible act of division, and it starts with the coiling of the chromatin to form chromosomes. As the coiling continues, the chromatin threads form into the conspicuous double chromosomes. Each set of identical twins is joined at a single point called a centromere. Now the nuclear membrane dissolves away. While all this is going on, an amazing structure of fibers begins to appear. They're known as spindle threads, and they run from one side of the cell to the other. A spindle thread is anchored to each centromere. You can imagine the spindle network as a kind of scaffolding on which the division will take place. Before we go on to the next phase, let's look at the prophase as it really takes place in a living plant cell. You can see the chromatin forming chromosomes. the nuclear membrane dissolves, and the cytoplasm and nuclear material freely intermingle. The fine threads are the spindle fibers. They're not easy to see in a living cell. Now the nucleoli disappear. At the same time, the twin chromosomes unwind. But they're still held together by the centromere. Up to now, the chromosomes have been scattered at random in the central part of the cell. In the next stage, the metaphase, they line up on the equator, ready for the division. The lineup is at a right angle to the spindle, and at the midway point between the two sides. The next stage, the anaphase, is actually the climax of the mitosis. Now, each centromere splits in half, and the two daughter chromosomes separate. Completely independent of one another, they will move apart. They're apparently pulled by the spindle fibers attached to the centromeres. The two identical sets of chromosomes move to the opposite sides of the cell.
Here's a blood lily cell in metaphase. The spindle fibers are slowly aligning the chromosomes along the equator of the cell. Now the anaphase. The daughter chromosomes separate, the spindle fibers pulling them to opposite sides of the cell. Here it is again. The chromosomes line up down the center. And the daughter chromosomes separate.
The final act of division is the telophase. In plant cells like this one, a cell wall begins to form, cutting through the spindle structure. The spindle fibers, guiding structures during the division process, now begin to disappear. While the cell is being divided in two, the chromosomes resume their original thread-like appearance, and new nuclear membranes form around them. Here's the actual telophase taking place. The cell wall begins to form, and the spindle disappears.
Now these are animal heart cells. You can see the duplicate sets of chromosomes emerging. The three dark nucleoli disappear. And the nuclear membrane dissolves. The chromosomes now line up down the center of the cell. Next, the anaphase, and the daughter chromosomes separate, each set moving to the opposite sides of the cell. In an animal cell, remember, there's no cell wall to cut the cell in two. Instead, the two daughter cells pinch together and pull apart. As the two separate, eruptions can be seen around the cell membrane. These are the result of physical and chemical adjustments that have to be made in the two new cells. Finally, the chromosomes mass together in each new nucleus, and mitosis is complete. This series of scanning electron micrographs shows an animal cell in division. The time required for such a division varies, depending upon the kind of cell. Some single-celled organisms can divide in less than 1 hour. Human skin cells divide in approximately 10 hours, and human brain cells don't divide at all.
Over 1 million different kinds of organisms have been described, each unique unto itself. Yet all of these organisms have basic characteristics in common. All organisms are composed of cells. These cells have many parts in common, and each common part has a similar function. These cells are produced from existing cells by mitosis, or cell division. Because of this cell division, organisms can grow and repair themselves, and life continues.